Welcome to SS2 Agricultural Science, Term 3, Week 3. Agricultural biotechnology uses biological techniques, including genetic engineering, molecular markers, and genomics, to improve crops. It represents the cutting edge of modern agriculture and is essential knowledge for any student of agricultural science. By the end of this lesson, you will define biotechnology and its branches, distinguish between traditional plant breeding and modern biotechnology, explain genetic engineering and GMO development, evaluate arguments for and against GMOs, state Nigerian agricultural biotechnology policy, and answer WAEC examination questions. Let us begin. Biotechnology is the use of biological systems, living organisms, or their products to develop or make products or processes for specific use. Agricultural biotechnology encompasses traditional biotechnology, fermentation, production of beverages, cheese, bread using microorganisms, conventional plant breeding, selection of desirable traits over many generations. Modern biotechnology, tissue culture and micropropagation, previous lesson, genetic engineering, recombinant DNA technology, molecular marker-assisted selection, genomics and bioinformatics. Traditional plant breeding, classical improvement farmers and scientists select individual plants with desirable traits, high yield, pest resistance, drought tolerance, and cross-pollinate them with plants having other desirable traits. Offspring are evaluated and superior type selected, repeated over many generations, takes 10 to 15 years to develop a new variety. Limited to crossing within the same or closely related species, gene pool limited. Advantages: natural process, consumer acceptance, well-established techniques, lower regulatory hurdles. Limitations: slow, many generations, limited to related species, cannot transfer genes across kingdoms, e.g. from bacteria to plants. Modern plant breeding, molecular breeding uses molecular markers, short DNA sequences that are linked to genes of interest, to select plants in the laboratory without waiting for the trait to be expressed, dramatically speeds up breeding. Marker-assisted selection, MAS, does not require the introduction of foreign genes, non-GMO, used in Nigeria for cassava, cowpea, sorghum, and maize improvement. Genetic engineering, recombinant DNA technology, the direct manipulation of DNA to introduce, remove, or modify specific genes. A genetically modified organism, GMO, contains DNA that has been altered using genetic engineering techniques. It may contain genes from a completely different species. Steps in creating a GMO plant. Step 1. Identification of the desired gene, e.g. the Bt gene from the soil bacterium Bacillus thuringiensis, produces a protein toxic to certain insects. Step 2. Isolation of the gene, cut out using restriction enzymes, molecular scissors that cut DNA at specific sequences. Step 3. Insertion into a vector. The gene is inserted into a plasmid, small circular DNA, from a bacterium, usually Agrobacterium tumefaciens, or coated onto tiny gold particles. Step 4. Transformation of plant cells. The vector introduces the gene into plant cells. Agrobacterium naturally infects plant cells and can carry the gene of interest into the plant genome. Alternatively, a gene gun shoots gold particles coated with DNA into plant cells. Step 5. Selection and regeneration. Plant cells that have successfully incorporated the gene are selected using antibiotic or herbicide resistance markers. Selected cells are grown using tissue culture into complete plants. Step 6. Evaluation. The transgenic plants are tested for trait expression, yield, and safety in the laboratory, greenhouse, and field. Examples of GMO crops. Bt cotton contains the Bt gene, produces a protein toxic to bollworm and pink bollworm. Cotton pest significantly reduces pesticide use, widely grown in India, China, the USA, and now being introduced in Nigeria. Bt cowpea, a cowpea variety engineered by IITA with Bt gene to resist pod borer, Maruka, approved for commercial cultivation in Nigeria in 2020, the first approved GMO food crop in Nigeria. Herbicide-tolerant crops, e.g. Roundup Ready soybeans resistant to glyphosate herbicide, farmer can spray the whole field to kill weeds without harming the crop. Golden rice, Rice engineered to produce beta-carotene, vitamin A precursor, developed to address vitamin A deficiency in rice-dependent populations. Potential benefits of GMO crops: higher yields, pest-resistant crops lose less to insects, herbicide-tolerant crops have less weed competition. Reduce pesticide use, Bt crops reduce insecticide applications significantly. Drought tolerance, genes from drought-tolerant organisms can be incorporated to help crops survive dry periods, important in Nigeria's savanna zone. Nutritional improvement, golden rice, vitamin A, crops with improved protein or amino acid profiles. Longer shelf life, tomatoes engineered for slower ripening. Disease resistance, resistance to viral, bacterial, and fungal diseases. Environmental benefits, fewer pesticide applications reduces non-target organism exposure, conservation tillage possible with herbicide-tolerant crops. Controversies and concerns, health concerns. Some people worry that GMOs may cause allergies or other health effects. The scientific consensus of major health organizations, WHO, FAO, National Academies of Sciences, is that approved GMO foods currently on the market are as safe as their conventional counterparts. However, some advocate for more long-term research. Environmental concerns, gene flow, transgenes may spread to wild relatives, particularly concerning in Africa where many crops have wild relatives, effects on non-target organisms, biodiversity, development of resistance in target pests over time, herbicide-resistant weeds, superweeds, developing in response to herbicide-tolerant crops. Socioeconomic concerns, corporate ownership of patented seeds, Monsanto/Bayer, farmer dependence on multinational companies, affordability for small-scale African farmers, intellectual property issues. Ethical/religious concerns, playing God, crossing species boundaries, religious objections to consuming genes from prohibited organisms, e.g. pig genes in plants in Islamic communities. Nigerian policy on GMOs. Nigeria enacted the National Biosafety Management Act in 2015 and established the National Biosafety Management Agency, NBMA, to regulate GMOs. The BT-copy approval, 2020, by NBMA was a landmark. Other GMO crops in the pipeline in Nigeria include GM cassava, maize, and sorghum for drought tolerance. Molecular markers and marker-assisted selection, MAS uses DNA markers located near or within genes of interest to identify plants carrying desirable genes without waiting for the trait to appear. More accurate and faster than conventional selection. Used extensively in Nigeria for cassava, resistance to cassava mosaic disease, cowpea, resistance to striga, witchweed, a parasitic plant, maize, a drought tolerance, WIMA project, water-efficient maize for Africa. Genomics and bioinformatics, sequencing the entire genome of crop plants allows identification of all genes and their functions. CRISPR-Cas9, gene editing, a revolutionary new tool that can precisely edit specific genes in a plant without introducing foreign DNA. The plant is edited not transgenic. CRISPR may face fewer regulatory hurdles than traditional GMOs since it can make changes indistinguishable from natural mutations. Already used to develop disease-resistant wheat, drought-tolerant crops, crops with improved quality. Biofertilizers, beneficial microorganisms, rhizobium for nitrogen fixation in legumes, mycorrhizal fungi for improved phosphorus uptake, applied to crops as alternatives to chemical fertilizers. Biopesticides, microbial pesticides, Bt, Trichoderma, Boviria bachiana, safer alternatives to chemical pesticides, organic certified, already used in sustainable agriculture programs in Nigeria. Key points: biotechnology uses biological systems for crop improvement. Traditional breeding, cross-pollination, selection, 10 to 15 years, limited to related species. Modern biotechnology, tissue culture, molecular markers, genetic engineering, GMO. Genetic engineering steps: identify gene, isolate, insert into vector, transform plant cells, select, regenerate, evaluate. GMO examples: Bt cotton/cowpea, insect resistance, herbicide tolerant soybeans, golden rice, vitamin A. Benefits: higher yields, reduced pesticide use, drought tolerance, nutritional improvement. Concerns: health safety, scientific consensus, approved GMOs are safe, environmental, gene flow, biodiversity, socioeconomic, corporate control, ethical. NBMA regulates GMOs in Nigeria, BECE-approved 2020. Molecular marker-assisted selection, MAS faster, non-GMO breeding using DNA markers. CRISPR-Cas9, precise gene editing, the future of crop improvement. Biofertilizers and biopesticides, biological alternatives to chemicals. WAEC test, define biotechnology, distinguish traditional versus modern breeding, steps in GMO development, examples of GMO crops, benefits and concerns, Nigerian regulatory context.